Immunology Insights, Basics and Beyond, Chapter 34, Type 1 Diabetes, Autoimmunity and Insulin Deficiency. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our previous chapter, we discussed rheumatoid arthritis, a chronic autoimmune disease primarily affecting the joints. Today, we will delve into type 1 diabetes or T1D, a prime example of organ-specific autoimmunity where the pancreas becomes the target. Unlike rheumatoid arthritis or lupus, which affect multiple systems or joints, T1D specifically targets the insulin-producing beta cells in the pancreas. So let's talk science. What is type 1 diabetes? Well, T1D is an autoimmune disease where the immune system mistakenly attacks the insulin-producing beta cells of the pancreas, resulting in insulin deficiency and hyperglycemia, which means high blood glucose levels. T1D usually manifests in childhood or adolescence, although it can occur at any age. The disease is typically diagnosed before the age of 30, with a peak incidence between 10 and 14 years. It is also sometimes referred to as juvenile diabetes because of its early onset. It is important to distinguish type 1 diabetes from type 2 diabetes. In type 2 diabetes, the problem is insulin resistance rather than deficiency, meaning the body produces insulin, but the cells do not respond effectively. Type 1 diabetes, in contrast, is characterized by an absolute lack of insulin due to beta cell destruction. It is noteworthy that type 2 diabetes usually is an acquired disease resulting from the lifestyle, while type 1 diabetes has a strong genetic component. Without insulin, glucose cannot enter the cells to be used for energy, leading to high blood sugar levels. This can result in acute complications like diabetic ketoacidosis or DKA, and long-term effects like cardiovascular disease, neuropathy, nephropathy, and retinopathy. Let's have a look at the mechanisms of autoimmunity in type 1 diabetes. The autoimmune attack in T1D primarily involves autoreactive T cells, targeting beta cells in the islets of Langerhans. These T cells recognize islets antigens as foreign, initiating an inflammatory response that leads to beta cell apoptosis. Let's look at the different elements. First, we have the T cell activation. The autoimmune response is predominantly mediated by CD8 positive cytotoxic T cells that recognize beta cell antigens such as GAD65, so glutamic acid decarboxylase, and IA2, which means insulinoma associated protein 2, presented by MHC class I molecules. These cells secrete pro-inflammatory cytokines like interferon gamma and TNF alpha, which directly damage beta cells. Second, we have autoantibodies. Patients often develop autoantibodies against beta cell antigens, which serve as biomarkers rather than direct mediators of damage. The most commonly detected autoantibodies are anti-insulin antibodies that appear early in the disease course, anti-GAD65 antibodies that are associated with longer disease progression, anti-IA2 antibodies that are often present at diagnosis. Last but not least, zinc transporter 8 or ZNT8 antibodies. They were recently identified as another significant marker. These autoantibodies are useful in predicting the onset of type 1 diabetes, particularly in genetically predisposed individuals. So, what are triggers and risk factors? As said before, T1D has a strong genetic factor. Genetic susceptibility is strongly linked to HLA class II alleles, particularly HLA-DR3 and HLA-DR4. These molecules present beta cell antigens to autoreactive T cells, enhancing the risk of an autoimmune attack. Additionally, polymorphisms in the INS gene that encodes for insulin and PTPN22, the gene affecting the T cell activation, also increase the susceptibility. But there are also environmental triggers. Several viral infections are hypothesized to act as environmental triggers. Coxsackie virus B is particularly implicated as it shares epitopes with beta cell proteins, potentially inducing molecular mimicry. Other possible triggers include enteroviruses, rubella, and cytomegalovirus, so CMV. Environmental factors like early exposure to cow's milk protein, low vitamin D levels, and stress, have also been investigated, but evidence remains inconclusive. What are the symptoms and consequences of type 1 diabetes? The onset of T1D can be acute or gradual, but symptoms often appear rapidly once a critical mass of beta cells is destroyed. First, we have polyuria, frequent urination due to osmotic diuresis from high blood glucose. Second, we have polydipsia, excessive thirst due to dehydration from fluid loss. Third, we have polyphagia, increased hunger despite eating due to cells being starved of glucose. Fourth, weight loss. The body breaks down fat and muscle for energy when glucose is unavailable. Fifth, fatigue and weakness due to the lack of cellular energy. And sixth, we have the diabetic ketoacidosis or DKA. It's a life-threatening complication where the body produces high levels of ketones as an alternative energy source, leading to metabolic acidosis. T1D significantly impacts daily life, as patients must manage insulin therapy, monitor blood glucose levels, and adjust their diet regularly. Uncontrolled T1D can lead to long-term complications affecting the heart, kidneys, eyes, and nerves. So how can type 1 diabetes be treated and managed? Managing T1D requires a comprehensive approach to maintain stable blood glucose level. First, there is the insulin replacement therapy. Multiple daily injections or continuous subcutaneous insulin infusions via insulin pumps are required. Different types of insulin are used, like rapid-acting, long-acting, and intermediate-acting, to mimic natural insulin release. Second, of course, blood glucose monitoring. Frequent self-monitoring using glucometers or continuous glucose monitors is required. HbA1c measurements reflect average glucose level over 2 to 3 months. Third, dietary adjustments are needed. Carbohydrate counting to accurately dose insulin is required. And of course, balancing macronutrients intake to avoid glucose spikes. And fourth, we have some emerging and promising therapies. We have immunotherapy, which explores ways to preserve remaining beta cells by modulating the immune response. Then we have Langerhans islet cell transplantation, an experimental approach aimed at restoring insulin production. And even artificial pancreas systems are explored, basically combining continuous glucose monitors and insulin pumps with algorithms for an automated insulin delivery. To conclude, type 1 diabetes serves as a key example for organ-specific autoimmunity, where the immune system specifically targets beta cells, resulting in lifelong insulin dependency. Its early onset and severe consequences make it a high-priority condition for early diagnosis and effective management. Understanding T1D helps us grasp the broader concept of how autoimmunity can target specific organs, contrasting with more systemic diseases like lupus. In the next chapter, we will step away from the disease mechanisms to explore a historical figure who laid the foundation for autoimmunity research, Noel Rose, the pioneer who first uncovered the concept of the immune system attacking itself. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.